Before I jump right into this, thank you for having me out here to the organizers. It's going to be pretty heavy downstream emphasis, but I think that you'll see that the combination of Genentech Roche families now partnering, that there's a lot of opportunities here for single-use implementation, so I'll go over just a few of those, and I'll give a little bit of background as well on those technologies. So I want to talk about disposable introductions in general, and these are not specific to downstream. They can be for upstream or downstream, especially when it comes to implementation strategies or testing, areas for use and some of the designs, as well as what are the strategies for implementation, what are some of the compliance testing, what are some of the snafus or bottlenecks you may need to deal with the quality and compliance organizations, as well as a case study that I'm currently leading globally for Roche and Genentech for implementation. So we all know, and we've seen today, some examples for end-to-end applications. Those applications offer a lot of opportunities. Specifically today, I want to speak about downstream systems that are usually in stainless steel, but now we're starting to see some very good ROIs, some very good NPVs, and very good examples and rationale for moving to disposable applications versus steel. Some of those things that you normally would move away from when you go to disposable, for example, are integrity testing, passivation, continued upkeep, preventive maintenance, scratches, dents, shipping, and validation. I will speak specifically to that in the case study. As well as we know a number of man hours are basically saved by using disposable implementation. For this crowd, we normally would talk about cell culture spinners and subs. However, there's actually even a lot more opportunities for downstream savings with disposables as well as upstream, but downstream is very applicable. So why use single-use technology? Your clean in place, your WFI generation, and your cost, as well as your clean steam generation is dramatically reduced when you start implementing at disposable. And I'm not talking necessarily at bench, I'm talking at full scale clinical and commercial GMP operations. So single use can dramatically reduce your operational complexity, your automation, which then can translate to headcount and staffing reductions as well as training reductions. And you can modify your facility footprint very quickly without cutting steel and having to go through new IOQ and passivations. All of those can lead to improved quality, specifically for aseptic practices where you get a gamma radiated system in. And that sterility then assures you a lack of bioburden. In cell cultures, it's very obvious. If you have a bioburden, you have a bloom and you know it. Downstream, you may not know it until you have your C of A and you have a frozen bulk, because it takes 5-day reads. And if you're at a 48-day run rate, you could have a couple of frozen bulks down the pipe before you know you have a problem. And all those benefits will transmit to financial benefit for the company's bottom line. So this is a couple years old, however, it's still relevant. Eric Langer in the United States does a survey, if you haven't seen it, and you're looking at scaling up or building new facilities, I definitely recommend it. And it's a survey of the top 100 biotech and pharma companies. And this is the output from, it's about a 180-page book talking about upstream and downstream and final fill. And I pulled out some facts from it. So currently, greater than almost 50% of the MABs are exceeding 1.5, 30% are almost up to 5 grams per liter now. And what that means is, as we have a cell culture conference, is the capacity and the constraint and the bottleneck is going to move downstream. So specifically for disposables, that becomes a challenge because plastic at large scale presents unique challenges, not to mention safety challenges. So what you want to look at, because of that, in the survey he asked manufacturers, directors, VPs, what are some of the constraints that you see coming forward in your production line? And these are what they listed as their top constraints. Downstream purification, better introduction of performing disposables, more cost-effective disposables, And the single most critical reason to use the disposables per the survey was eliminate the cleanings and reduce capital investment, which I've gone over just a minute ago. So here's just a review of some bags. We call these 2D bags, and these are general. You probably use a lot of these in cell culture, so high clone medias come in. You could batch your own medias in them. JRH sells medias in them. LifeTech sells medias in them. In general, they're hanging bags. They don't require external 3D container. When you start getting to larger applications, then you require an external container. And while it may seem mundane and routine and arbitrary to be talking about this type of stuff, when you start talking about large-scale GMP, engineering, and movement, an example was when I was working in the Amgen facility in Rhode Island, the facility had to actually be re-engineered for the run rates to handle the totes, because we had over 180 of these totes, which are about 4 by 4, and you start thinking about where you store those, ergonomic considerations of moving those, and a lot of those were for a perfusion process for Enbrel. So while it may seem obvious, how to stack and store and cool and warm these is not obvious. And you need to consider that when you're talking about a scale up from bench to full scale. And of course, we all know about sampling bags. And while it may also seem obvious, sampling bags are nice. In this example, here's Novaseptic from millipores bags, and I have some charter-mounted sampling bags here as well. You have to plan in your implementation disposables such things as taking LAL samples. You can't take LAL samples in these bags. They actually absorb the LAL and you'll get false positives. So thinking about your implementation strategy is important with the use of disposables. It may not always seem obvious. So single-use applications are meeting our needs as we see them and helping us debottleneck. However, the coordination of the data and the use across large organizations such as Genentech and Roche can be quite complex. And the reason it can be quite complex is that if you have multiple filings and multiple buildings, multiple states or countries, understanding what are in these raw materials as you use them, and here we use an example of media and buffer prep, and this is a high clone system, thermo high clone system, understanding what that backbone is, understanding where they got their rolled film from, where they do their blown extrusion from, and what the C of A is, and even if you have that information on the C of A, such if there's a change control, you may not even know it. So if you move back for a minute and you think about the raw material, the plastic bead comes from Dow Corning, or Mobil, or Exxon. These are very large corporations. The plastic that they make for biotech and pharma is an extremely, extremely small slice on the pie of their plastics. And so their change control process may not be what you think it is going to the thermos and SSBs and millipores, et cetera. So understanding and coordinating your raw materials and changes that can then, and I don't know if people have heard some of the studies and stories about films having an effect on cell growth. It's been well characterized and published now. So there are some films that can affect cell growth deleteriously, not to mention extractable, leachable profiles. Here's some examples. I just showed media. We've got disposable spinners now. Here's an example that we were demoing a year ago in the lab where we were doing closed tubing sets. So you wouldn't have to do any of the transfers under laminar flow. You could just do them tube welding. And of course, we get very large with a 500-liter wave bag here. I've seen these, and you certainly don't want to be on the side of this that have ever rolled towards you. So that, I think we're moving away from 500-liter bags into stirred single-use bioreactors now. But I just had to show that because it's a very large bag. I'm going to show a full disposable application in a second. Here's examples of how you make up that application. Here's the harvest system. So here's one from Paul. Here's one from Millipore. Both of them work very well. We've used them as well in our clinical operations at phase 1 and phase 2. And they have very high throughput, and they negate the need for centrifugation or cleaning because there are no wetted surfaces that you have to basically clean or perform coupon studies on. All the wetted surfaces are closed and disposable. So we start moving into the larger reactors. Here's an example of 250-liter sub. This is an old picture of it, but it basically is the same idea. Sartorius has a similar application. And then near and dear to me, my role is downstream manager for manufacturing sciences for our Oceanside commercial manufacturing facility, or downstream applications. So this is ready to process prepacked columns. I was talking during lunch with a couple of folks who were asking, why would you want prepacked columns? A lot of energy goes into packing columns, and failed efforts go into packing columns, as well as the validation around the HDPs and the training of operators. If you have a pre-packed column, you move away from cleaning, setup, regeneration, and storage of the actual systems, and you plug these in with these disposable connectors. I'm showing these here because they're agenderless, which are very nice. So if you have a system and you change the order that you're using these downstream applications in, you don't have to worry about changing your engineering diagram and creating a new material specification. So that's thinking about your implementation and where you're going to use these applications are critical, and using an application such as this that doesn't have a male and female end allows you to interchange them easily. So the things that may seem intuitive will get you in the end as far as your timelines go if you don't think about those connections. Here's an example of one of the first fully disposable chrome skids. While it has stainless steel on it, you see the pinch points that the fluid goes through the tubing, and the pinch clamps here, the pinch valves, then open or close the pathways depending whether you want to do a load or a bypass or an elution to the column. And then this runs off a unicorn system. So very nice. It's limited to its flows and speeds and pressures, obviously. So it would not be much past phase 2. This is, as I'm aware of, the very first disposable UFDF skid. We designed it about 3 years ago. Now there's commercially available ones from Novacep and a couple other competitors out there. Millipore, Sartorius makes one as well. But we designed this for ADCs, for antibody drug conjugates. And so basically the whole point behind this wasn't to save labor, but was protect the employee. So what we have here is a fully disposable flow path so that you don't have to clean any of the dangerous conjugates. So you just do your conjugations in a closed system and then throw them away. That was the idea. Now they're commercially available. Oops. So some things to think about. This is just an example of disposable mix system by Millipore, but these are some of the areas I was talking about. You could have all different type of connectors here, and this disposable mix system could be not just a mix system. This could be your load tank to a column. This could be a pool tank. It could sit on load cells. So there's a number of ways to slice the pie here and use one system to meet many needs. It could also be a harvest tank at basically a small scale. Let's say up to 100, 200 liters. These are perfect harvest tanks that you then use in multiple configurations for the load tank and the pool tanks from the chromatography unit operations. And then depending on your connections, you can use it interchangeably in each of the unit operations. So we talked about unit operations, same unit operation cost. That's one driver. But there are other drivers as well. And so I want to use a case study here that I'm currently working on, and I'll be meeting in Basel with the folks next Monday on this. We're looking at replacing our stainless steel cryo vessel. So like Amgen, Genentech, Biogen, and a number of other companies that I'm not aware of, but there's some of the companies I have worked at. These are basically the gold standard for freezing API. So you do your protein purification, you put your bulks into the stainless steel freeze tanks, and then you have what's called a cryotoll or controlled freeze unit that then freezes these. Well, these tanks are about $175,000 a piece. I think Genentech has somewhere around 1,800 of them just in the US. And then you have to do PMs on them. They have to be cleaned, they have to be steamed, you have to do visual inspections. There is a small community, no joke, around these just managing just the tanks. So something that we did when I first came to the company was we introduced a disposable freeze bag, which may seem simple, but it's not. You have to look at things like the glass transition of the plastic. Is it able to take -50? Do you have transportation studies for it? Do you have freeze studies for it? Have you gone through your local municipality? Do you have international shipping protocols for it? And will it withstand droppage and breakage? So we basically manufacture all of a clinical scale phase 1, 2 materials in these, and we have designed a system with thermo that does a control rate freeze through Delta V, controls the freezer, and also does builder management system and looks at the rate of your freezing, so we don't have to use these stainless steel tanks anymore. Well, that's only good to a certain level. So you start freezing 300 liters at a time, you're not going to want to be filling 15, 20 liter bags. Here's a disposable shipping system that we engineered, partnered with Nanjing Nauk and Thermo to come up with a disposable packaging system. So basically everything is one way, there's no validation, you don't have to clean it, wipe it down, or bring it back, and it goes to the fill site. And then this can be wiped down and go into clean 10,000 particle space for fill. So our paradigm was, how do we move out of the large scale for commercial production versus those small bags and get into a large fill system that allows us to fill, in this case, 1 to 300 liter fills for an average MAB of between 12 and 50 grams per liter, 20 to 30 kg per run. So one option, and this was developed internally at Genentech from Philippe Lamb's group, was to put together a competing tote versus that other freeze tank that would be reusable, and there's no product contact. You put your bags in here, and you freeze your bulk, and you hook this up to the cryotrol. However, you still have to maintain this unit. It has a chiller, and you've got to ship the stainless steel cross-country and still have a team that manages. And with just-in-time manufacturing, you can't necessarily have an endless supply of these at all the manufacturing plants. It's very not cost-effective. Here's just an example of showing the testing we did, so you can basically see the freeze fronts, the last point to freeze, and to monitor your rate of freeze and your last point to freeze. So what my team did was put together a business case for the global Roche Genentech bulk freeze networks and look at the amount of resources required for labor, for setup, for shipment, and all the safeguards that are put together for the risks and shipping, cleaning, and validating large tank management centers and the PMs, and put together a project to replace the stainless steel tanks. So the background and the scope was to assess the commercial drivers, assess maturity that exists in the external marketplace already versus our internal tank that I already showed you that was developed, and align on one freeze system globally, define the user required specifications and the drivers for international alignment globally, and then look at those one-way logistics in the network and review them. Here's what the mission milestone plan looks like. We have 3 milestones. We're currently in milestone 2. So we've already had our business development, approval, and assessment and maturity, and I'll show you the final system we aligned on in just a second. We have already executed with Sartorius Tedim at their sites in Göttingen the commercial system that we have agreed upon, developed procedures, and now we're doing stability and freeze studies on site in San Francisco. Then we'll move to the at-scale feasibility and transfer stage for our first commercial product. So here again are the drivers. So long-term storage of steel, depleted cryo vessels, handling, cost of quality, and cost of quality is actually a large driver. And the reason it's a large driver is when you fill one of those stainless steel freeze tanks, they have small sight glasses on them. So if there's something, which I'll show you in a second, if there's particulates or a foreign object in there, you don't know it until after you've filled the bulk drug into vials. So then when you pull the tank out and you lift up the head and it's in a different tank management center and you find something in there, all of your fills are then implicated. Other drivers then are as complex tracking systems, scheduling, cleaning validations. You have to do large retrofits to handle these tanks and to manage them globally. Not to mention the labor and expenses just for clean in place SIPing as well as manually manipulating the steel is a very high user point. And they're very heavy. And we've had ergonomic issues for sprain, strains, and pinches with these large tanks. So with the disposable implementation, we believe that we can reduce cleaning, no cleaning validation at all. There should be no reprocessing doing the rouge, debris, scratches, or any other repairs, which I'll show you examples in a minute. Reduce cost of quality because of deviations and/or foreign objects found in the stainless steel tanks. And obviously a reduced amount of capital because we're not going to be purchasing, as a company grows, more stainless steel tanks and lower cost of goods, which I'll show you examples of in a minute. And we shouldn't have these occurring. Here's an example. It's obviously an extreme example, but here's a large tank, and you can see they're quite large and need forklifts to move them around. And so with our new system, we believe these type of examples should be eliminated. Here's an example of visual inspections that occur in these freeze tanks, and you can see they're large systems. They have to have pick points. The fins have very small clearance between the sides of the walls, and they can cause scratches when you lift them. And these are also in non-classified areas. So could we do this in classified areas? Sure. But then we increase our cost even more, because then you got to pay for air handlers. But this guy's hat could fall in there. This guy's wrench could fall in there. This little yellow twist tie or something sitting on the edge could fall in there. It's possible we found those things. I wouldn't say if we did, but you wouldn't find those things because there's no assay for these. When they fall in there and you do your clean in place and your SIP, if an operator looks through there, you won't see that. Here's examples of rouging, pitting, particulates, and this is the final bulk. This is it. So after it's gone through cell culture or harvest, purification, the bulk goes in here. If anything sheds from these tanks, if it does pass through a 0.2 filter and find a fill, it's going to go into the vial and go to the patient. And that's really what we're all about. This whole industry is about making unmet needs for the patient, and we certainly don't want that hitting the patient. More pictures of damaged spray rings, investigations around the spray rings, and validations for the patterns. This is a typical flow of a tank, and it's quite complex. And of course, I've talked about disposables for purification. This is just a freeze tank. So there's a lot of offers and applications for disposables. But there's a lot of steps, and you can see that at my bulk site in San Diego, we receive the tanks, and we got to prepare the tank. This takes a couple of days. We got to put pressure on it, make sure there's no leaks, perform the freeze. The fill sites have to receive it. And a lot of times they marry these together. So they may pull 5, 6, 7 tanks together from multiple runs, marry them, and then perform their fills. Then you got to manage the empty tanks through the fleets and then go back to the drug substance site. So there's a lot of, this simple chart has a lot of moving parts and a lot of costs associated with it. Here's an example of that. So here's all drug substance sites in our Genentech Roche network. Here's all the fill sites. So it looks quite complex, and I assure you it's more complex than just lines when it comes to getting the tanks to the right place and the right time for your manufacturing production plan, which is not static. And then you'll see that you have a return, and this really is quite a bit of energy and time and money spent on just returning the stainless steel tank. So we believe that if you go to a one-way disposable application to get rid of the stainless steel tank, for example, then you move to something like this. Get rid of the middle guy. So there's no more management and the return of stainless steel with a disposable application. Here's an example of what we believe are the biggest drivers. I'm sorry about the font. It didn't translate. I was using some Roche fonts, and they didn't translate on the PowerPoint. But invest, manufacturing robustness, quality, right to operate, design, engineering, and supply chain were the biggest drivers. And here are some measures that we got together as a team with a bunch of subject matter experts, and then ranked the different disposal applications. Here's how they ranked. So we looked at traditional cryo vessels, which are the stainless steel. The Baxter was our internal application that looked like the toaster with 3 slots. And then the external Sartorius Didim, because we scoured their environment. We didn't find anything for 300-liter fills that was ready for implementation. I think I could skip this for you. We've talked about pretty much everything here except the water reduction for steam and water. When you talk about cleaning and SIPing, there's a very big green utility savings by going disposable. And while it may not seem intuitive to throw a bunch of plastic into a landfill or do cogeneration, I know in Europe they do much more cogeneration and burn the plastic. It may seem terrible for environmentalists. It's actually more green to bury or burn plastic and throw it away than it is to deplete water and use fossil fuels energy to generate WFI and steam. So there's been a couple of publications from myself and a few others from Roche on that, and from Biopharm, from Andrew Sinclair. If you want to look that up, they're quite good papers as far as the argue for it. So here's our final application. This is what we're working on for implementing. It's a fully disposable solution. It's not an advertisement for Sartorius to deem. However, that is the system we are relying on. This casing is about as big as this podium right here. So it's a valid application for commercial implementation for bulk freeze. And you'll see there's a slot right here. There's a slot on either side, and what you're looking at is a top for tube works. There's a bag that goes inside of it, and then there's a top that goes on it. So while it's nice and pretty and it looks like a big waste, the full thing would be disposable. So we would fill the bag inside of it, perform the freeze, ship it off the drug product fill, and that would be the end. Because once you start to have to wipe it down, or to manage it, or ship it, or bring it back and figure out what do you need where, then it's the NPV and the ROI are severely diminished. Here's another look at what 3 of them together would look like. So that's a 300-liter fill, and the beauty of it is instead of having one 300-liter tank, you could have increments of 100. So if your SAP in your final drug product is calling for a 600-liter fill, let's say 500, you may thaw 5 of these instead of having to thaw a 300-liter bulk freeze tank and then refreeze 100 liters, which then there's issues with refreezing protein. You can only do it and refilter it so many times. Here's a rendering of the beta unit that we're working on currently, and then an example of the bag being lowered down into the unit, and then of course you can put whatever you want on the end. So you could tube weld, you can sterile filter, you could have quick disconnects, you could have any, Paul clean packs, whatever you decide on there. Here's a rendering of the beta unit and our teams performing the beta trials on it, and the slots right here, these are freeze fins that freeze silicone oil and move it through the freeze fins, and it goes down and sandwiches the bag and then performs a freeze across the fins, and that's a controlled freeze. The beauty of this is that in the majority of our bulk networks, we have the cryotrols already installed for the freeze tanks. Sartorius, making a good business decision, decided, hey, what do we have that we can implement in a disposable mode such that then the end user doesn't have to go and buy more capital? Well, we already have, as so do quite a few other people in the room, in their companies. I may not know it, they already have these cryotrols in their company. So this can fit right into that disposable application without adding a lot of capital. I was going to tell you how much it is, but lawyers may not like that. We're under contractual agreement. It's a collaboration with them right now. I think it is a positive return. Here's an example of what they look like. Here's a rendering. Here's the actual. They have specific pallets where these fit onto the pallet. And then they're clamped down, and they're quite difficult to turn over. Very robust, and a very nice system. And they have a new shipper for them. They can hold them at -20 for over 72 hours without any external electricity or any other energy. Dry ice blocks go up top, and it's ASTM certified for shipping internationally and domestically. So the realized benefits behind this, summarized, would be less capital, materials, and labor, reduced downtime because you're waiting for steel. We've actually had times where we're making bulk at some of our bulk plants where we have to slow down our run rate because we can't get the tanks back in time to clean them and fill them. And that can actually be a constraint in your bulk network. It would become a bill of materials, just like a filter, a bag, or anything else, and you should have those just in time in your warehouse at each of the bulk sites. Less water, less steam, less electricity used because you're not doing CIPs and SIPs. Allows for a higher level of innovation. It's more amenable to lean manufacturing. Allows for a quicker return on the supply chain. However, you do have to perform your extractable and leachables, and so I do have a few minutes here. I won't go into that too much. I can take some questions on that. That's an entire work stream in itself, obviously, especially at the bulk fill stage. So let me talk about that real quick. So I have a few minutes, right? Go over. Again, back to a coordinated strategic approach across a company is critical. Disposables are great, but to implement them, you need to assign a project manager, define the business scope. Where do you want the bioprocess container? What are the vendors you're going to use? What are the industry standards? So I lead ISPE for disposable applications for EU and the US. So we're developing guidelines and standards for what the testing and acceptance criteria should be for disposable applications, and working with ASTM and the PDA to generate those standards and white papers for review by the regulatory agencies. You want to develop a schedule, develop a project plan, and then conduct your technical assessments, and then deploy a long-term strategy. Such as, here is this is an extract from our comparability protocol. So Genentech submitted this to the FDA. This is the first that I know of where we said, this is how we're going to test our films. This can apply to bioreactors or the final fill. It's obviously more critical near the patient fill. However, I do believe that you cannot argue that if you use an SUB and you use a plastic bag in the SUB, that it's removed. So unless you have some pretty complex models showing that protein A binds and does not release those extractable leachables, I would probably say it's not reasonable to say that you don't have to test your films in upstream. But let's see what happens with standards. Do I want everyone to have to use Toxicon for testing? Not necessarily, but I think it's prudent to understand your films and have a good quality justification. Yes, so here is an extract from that paper we submitted to the FDA, and we said this is how we're going to qualify our films in general, whether they're used for cell culture or the final fill. So we perform material suitability, we have microbial integrity, and these are per CFRs or specifically ICH Q7. And then you perform your extractable studies, and then you do a tox evaluation based on what you fill. Each of these are procedures internally to Genentech and Roche that we have. However, I published this 2 years ago in American Pharmaceutical Review, and it goes into what the acceptance criteria are for each of these. And then we have internally at Genentech a tox evaluation based on the peaks that we find. Then you also want to look at the chemical compatibility, leachable studies, and then do an evaluation of both the chemical compatibility and the leachable studies against Q3C and the ICH guidelines, and look at those acceptance limits against your API, depending on the dose. Use a worst case scenario. And if it follows the flow sheet, if it passes everything, you're in good shape. If it doesn't, then you may have to do some retesting or perhaps not use it for a unit operation. We also do a health-based risk assessment depending on what drug we're using, what the extractables are, and what the toxicology assessment is against the ICH guidelines. And so this is just a flow chart. I won't go through it. But depending where you have a parenteral, an inhaled product, or an injectable, obviously, the guidelines for what's acceptable and those limits, average daily permissible limits, are different. And so these are part of our SOPs. And so the folks in the manufacturing sciences would do an assessment based on extractable profile, and will qualify a generic film. So if we qualify CX514, for example, from Hyclone, it's qualified, and it's qualified against a model solvent family for use. And we use the worst case scenario with the largest dose so that we can use it across our drugs. We don't claim it for a specific drug, which is probably a safe way to go. And we claim no removal from any steps. No UFDF, no protein A. We assume everything that extracts from an SUB is concentrated and goes into the final dose, which is obviously very conservative. However, we find that in the worst case, we're still about 0.05% of the allowable limit, which is very safe. So some definitions quickly for you. Class 1 extractables are possible carcinogens. They pose serious health risks, and if you found one, you probably would not want to use the plastic. Class 2 have some level of toxicity. They're suspected. They have structures that are similar, however not as toxic as the Class 1. They can be from 0.5 to 50, but they should be, where possible, avoided. And then Class 3, the majority of what you'll find in extractable leachable profiles from the vendors, from Sartorius or millipore steam, et cetera, hopefully fall within Class 3 extractables. A lot of the vendors have this information. However, it may not be the model solvents that you're using that you can bracket and validate and then assume that those are your solutions that you use in your process. So if they do have that information, you're good, and then you can reference that. But you also have to consider their standards and their testing. So were their standards and their testing the same as the guidelines that we're currently expecting within the regulations. So some reasons for testing is that the components must meet the required biocompatibility for use, for temperature, and chemical mechanical properties per FDA NICH to ensure that the chemicals are compatible with the plastics. While they're saying great, you still need to do your diligence. And as I said before, we have found some situations, most recently I've had a couple of talks with folks where there's limited cell growth because of the. Here's an example where, while it may seem trivial to implement a bag, if you want to look at the true implementation, and this is just for a buffer bag going into a commercial process, it took us about 230 days, if you include all the validation, testing, extractable, leachable, and approvals. So just an idea of where you want to go if you want to implement a commercial. And here's a strategy for doing a full-scale implementation. It's about 14 to 16 months with filings. I've covered everything, I believe. And I want to thank Sartorius team, specifically Jonathan Cutting, who's one of the engineers who developed that disposable solution. Okay, thank you.
Thank you very much.